Welcome to the Deep Dive, where we take your stack of sources, articles, and research and distill it down to the essential insights you need to know.
Hey, glad to be here.
Today, we are tackling one of the most stubborn hurdles in modern drug development, and that is the search for effective combination therapies.
It's an absolutely crucial topic. I mean, whether you're fighting cancer, which often develops resistance, or infectious diseases, combinations are really the standard of care now.
But finding those perfect pairings is notoriously slow and inefficient.
It is, and the research we're looking at today really highlights why it's so hard.
It's not just a lab problem, right? It's actually a math problem.
Yeah.
Our sources call it the quadratic bottleneck. For listeners who are, maybe getting up to speed on this, can you break that down for us?
Sure, so think about it this way. If you have, say, 100 drugs you want to test, that's 100 experiments. Simple. It scales linearly.
1 to 1. Makes sense.
But now, if you want to test every possible two-drug combination from those 100 compounds, you suddenly have almost 5,000 unique pairs. And if you go up to 1,000 compounds, which is a pretty reasonable number for a modern screen, the number of pairs just explodes. It's nearly 500,000.
500,000 combination experiments. You can't practically run that many screens when you factor in controls and replicates in different doses.
You can't. You run out of budget, you run out of time, and you run out of the actual drug compound, which can be incredibly precious.
So this quadratic increase just shatters the whole idea of doing a comprehensive screen.
Exactly. We know the key combinations are out there, but we've been forced to rely on educated guesses or really small focused screens or just pure luck.
Okay, so that's the monumental challenge. The research introduces a pretty radical solution to this, Combocat.
Yeah.
It's an end-to-end open source platform. What's the big picture here? How does Combocat claim to shatter this bottleneck?
It's a really smart, integrated approach. Combocat marries cutting-edge hardware, specifically acoustic liquid handling robots, with predictive software, sophisticated machine learning.
So it's not just a better robot or a better algorithm, it's the two working together.
It's a unified strategy. By making the experiments ultra-precise and then using computation to fill in the missing, say, 90% of the data, it achieves what they call ultra-high throughput.
So our mission today is to really unpack how this all works. We'll look at Combocat's two modes, this high-fidelity dense mode and the super-efficient sparse mode, and we'll see how they were used to screen a record number of drug pairs.
And to really start, we have to define the goal here, which is finding synergy. We can't talk about how it screens combinations unless we're clear on what makes a combination valuable in the first place.
Right. When most of us say synergy, we just mean working better together. But in pharmacology, it has to be way more rigorous than that.
Absolutely. Synergy is defined as a combination effect that significantly exceeds the expected additive effect. And this is based on established models, most commonly the bliss independence model.
Okay, so walk us through a simple example of bliss independence. What does that look like?
Okay, so imagine drug A by itself kills 20% of the cancer cells. And drug B by itself kills 30% of the cells. You don't just add them up to get 50%.
Right, because some cells might have been killed by both drugs anyway.
Exactly. The Bliss model accounts for that overlap. It would predict the combined kill rate should be 44%.
So if your actual experiment shows the combination kills, say, 70% of the cells, that extra 26%, that's the synergistic boost.
That is synergy, and that boost is vital. It means you can use much lower doses of each drug.
And lower doses mean less toxicity for the patient. And it can also help overcome drug resistance.
Precisely.
But historically, finding that sweet spot was so hard because of what our sources call the dose-density trade-off. Researchers had to choose, test lots of combinations with few data points, or test a few combinations with lots of data points.
Right. You'd see these small, like 3x3 matrices, or even weird asymmetric ones like 2x7. You just miss the nuance. You can't see the full picture of the dose response.
And this is where the hardware solution comes in. Acoustic liquid handling. We're talking about mind-blowing precision here, aren't we?
We are. We're talking about nanoliters, a billionth of a liter. This tech uses focused sound waves to just pop these tiny, precise droplets of compound from one plate to another.
without any physical contact.
No contact at all, no cross-contamination, and it's incredibly fast. And that precision enabled Combocat's first mode, the dense mode workflow.
So this is the foundation. Dense mode is all about getting the highest quality data possible.
That's right. It systematically measures all the pairwise dose combinations in a high-resolution, dense 10 by 10 matrix. That's 100 unique concentration pairs for every single combination.
And they do this in triplicate, right? With a ton of internal controls built right into the plate.
Exactly. You've got 12 replicates each for your cell death and vehicle controls, so the normalization is really robust.
So once you have the raw data, it goes into this analytical pipeline. And after mapping and normalization, there's this really critical step, quality control. Why is that so crucial here? Can't you just trust the robots?
Well, the robots are phenomenal, but they're not perfect. You might get an incomplete drug transfer, maybe a bubble messes with a reading. These create what they call spurious single-agent measurements. Outliers.
And if your baseline measurement for a single drug is wrong, then your whole synergy score is going to be thrown off.
It'll be completely corrupted. So Combocat has this sophisticated QC system that flags these technical hiccups before calculating the final score. It looks for three things.
What are they?
It checks for variability between the replicates. It looks at the residuals, how far a data point is from the fitted curve. And it checks for monotonicity.
Monotonicity.
Okay.
It's just the idea that as you increase the dose, the effect cell death should also increase, or at least stay the same. It shouldn't go down.
That's it. If you see a huge drop in cell death when you triple the dose, you know something went wrong technically. So the pipeline flags that, adjusts the data, and gives you what they call adjusted bliss synergy, or bliss pristine data.
They use this dense mode to build a huge reference data set, right? Over 800 combinations. Did they validate it? Did they prove it works?
They did. They successfully replicated the famous synergy between two antibiotics, SMX and TMP, in E. coli. And in neuroblastoma cells, they validated a known combination there too. So it confirms, yes, dense mode gives you that reproducible ground truth data.
Okay, let's hit our first recap. We've just laid down the foundation that Combocat is built on.
Sounds good. So our recap 1, the foundation of fidelity. First, the big problem is the quadratic bottleneck, making comprehensive screening basically impossible.
Right.
Second, the dense mode solution uses acoustic liquid handling for these super high fidelity 10 by 10 matrices.
And third, the secret sauce is that rigorous quality control pipeline that filters out technical errors to generate a reliable, adjusted synergy score.
That's the bedrock of the whole system.
Okay, so dense mode gives us incredible high-resolution data, but it's still pretty resource intensive if you want to check thousands of pairs. So how do they get from high fidelity to ultra high throughput?
And that is the transition to the big breakthrough, sparse mode. This is where Combocat uses computation to solve the rest of the scaling problem.
The goal here is to do way fewer experiments, but still get the full picture. How much did they miniaturize things for sparse mode?
They pushed it to the extreme. They went from a 384 well plate to a 1536 well plate. The total volume is just 4 microliters of cells and only 20 nanoliters of compound. A massive saving.
20 nanoliters, that's almost nothing. So that's the hardware side. What about the mathematical trick? How do they cut the measurements by 90%?
So instead of measuring all 100 dose pairs in the matrix, sparse mode measures only the 10 diagonal dose pairs.
Diagonal. So that's where the two drugs are in a relative 1-to-1 ratio.
Exactly. They measure those 10 points plus the 10 single-agent responses for each drug. So you only measure 30 points in total, not 100 plus.
Okay, so you have the response curves for each drug alone, and you have this one diagonal slice of the combination. But you're missing 90% of the data. And this is where the machine learning engine comes roaring in.
This is it. This is the software solution. Combocat uses an ensemble ML model trained on all that pristine dense mode data to predict the 90 missing values. It mathematically reconstructs the entire 10 by 10 matrix.
An ensemble model. That sounds complicated. Why did they use 90 different models instead of just one big one?
This is a brilliant architectural choice. The ensemble is made of 90 individual XGBoost regression models, and each model is a specialist. It is trained to predict the cell death for one specific non-measured coordinate in that matrix.
Wait, so the model predicting the effect for, I don't know, the 3rd dose of drug A and the 5th dose of drug B is a totally unique model, trained just for that one point.
That's exactly right. It's incredible. They trained this whole specialist army on over 500 of those fully measured matrices from the dense mode reference set. So it learns the subtle curves of synergy from the best data possible.
And how good is it? I mean, can you really trust the 90 predicted points?
The accuracy was remarkable. The overall correlation between the real observed values and the predicted values was 0.98. The R-squared value was almost 0.95.
Which basically means we can trust this predicted data almost as much as if we had run the full experiment.
That's the takeaway. But here's the key insight that makes this more than just clever engineering. Feature proximity.
Okay, what does that mean? Did the models rely on all 30 measured values equally?
No. And this is where we learn something new. The analysis showed that the predictions were overwhelmingly influenced by the measured values that were physically closest to the target point on the matrix.
Closest. You mean in terms of Euclidean distance, like on the grid?
Exactly. The model doesn't use a measurement from the top right corner to predict a value in the bottom left. It uses the immediate neighbors. It confirms that the combination response is a locally coherent thing.
So the model works so well because the few points you did measure contain enough information to reliably guess what's happening in the gaps right next to them.
It's a scientific confirmation that this whole dedicated ensemble approach is the right way to do it. It's an elegant marriage of automation and computation.
All right, time for our second recap, this time focused on the scaling engine.
Okay, recap 2, the scaling solution, sparse mode and machine learning. First, the efficiency trick is miniaturization, 1536 well plates, and only measuring that diagonal, which cuts the workload by 90%.
Got it.
Second, the engine is that ensemble of 90 dedicated XGBoost models that reconstructs the missing data with incredibly high accuracy.
And finally, the key scientific insight is that the model's success relies on local features, feature proximity, which validates the whole approach.
And that's how you get to ultra-high throughput.
So, let's talk impact. How did this scaling ability translate into actual discovery? What did they do with it?
Well, they screened a massive 9,045 drug combinations in a neuroblastoma cell line. And this is the largest dense combination screen reported in a single cell line to date.
9,000 pairs. That is just a staggering number. But we know strong synergy is rare. So how did they filter through all that data to find the real hits?
They used 3 really rigorous filters. The first one was spatial coherence. They used a metric called Moran's eye to make sure the synergy scores weren't just random noise scattered across the matrix.
So Moran's eye is basically asking, is the synergy happening in a logical clustered block on the dose map?
Exactly. Real synergy should cluster. The second filter was data reliability. They threw out any matrix where the QC pipeline had flagged too many of the 30 measured points as being unreliable.
Makes sense. Garbage in, garbage out.
Right. And the third filter was biological relevance. They only kept matrices that actually achieved at least 20% mean cell death. You want synergy that actually does something meaningful.
So after all that filtering, what was the biggest discovery they found and then validated?
The standout hit was this really strong synergy between an ATM inhibitor called AZD1390 and PRP inhibitors, specifically olaparib and rucaparib.
And mechanistically, that makes perfect sense. You're hitting 2 complementary DNA damage repair pathways.
It's a beautiful 1-2 punch. PRP inhibitors cause a lot of single-strand DNA breaks, which then become lethal double-strand breaks. And then the ATM inhibitor comes in and stops the cell from repairing those double-strand breaks. The cell has no way out.
It's a powerful validation of the entire platform, but we do need to address the limitations and the caveats, which are really important for any listener trying to understand this.
Absolutely. So first, you have to recognize the technical shifts. Sparse mode uses different plates and smaller volumes, and that can sometimes cause minor shifts in a drug's apparent potency compared to dense mode.
Okay, that's a technical note.
Yeah.
What about on the scientific side?
A major caveat is the choice of the synergy metric itself. The platform supports both BLISS and LOW models, but the sparse format revealed a fundamental problem with the GLOW approach here.
What was the problem?
The LOW model requires you to measure specific dose-equivalent points, which, because of the diagonal-only measurement in sparse mode, just weren't there. They couldn't be measured.
So what did that mean in practice?
It meant that low synergy was mathematically undefined in 61.5% of the sparse mode combinations they tested.
Wow. Over 60% of the data couldn't even be evaluated with a major established synergy model.
It shows how much your experimental design limits the questions you can ask. It forces you to rely on the BLISS model, which is robust but still has its own assumptions.
Okay, let's wrap up by talking about the road ahead. This whole thing, the protocols, the code, it's all open source.
And that is the long-term game changer. By making it all freely available, they're creating a system for community-driven advancement. Any lab can generate new dense data and contribute it back.
And as that global database of high-fidelity synergy maps grows,
The machine learning model just gets retrained and gets smarter and smarter. The more data it sees, different cell lines, different drugs, the more accurate its predictions become for everyone.
So Combocat is this scalable, reproducible platform that really moves drug combination screening from this limited manual art toward ultra-high throughput discovery.
It's the infrastructure for a whole new age of systems pharmacology.
And that leads to our final provocative thought for you to consider. The source material emphasizes that strong synergy is a rare thing. It's often hiding in these massive data sets. So, if machine learning can now efficiently map these synergy landscapes across thousands of drugs and cell lines, which was just impossible before, which undiscovered conserved synergistic interactions might we find? Could we uncover entirely new vulnerabilities in diseases like neuroblastoma that were completely invisible in smaller screens?
That scalable, high-fidelity screening unlocks a whole new pharmacological universe to explore. That search for the invisible synergy, that is the mission that's now within our grasp.
Thank you for joining us on this deep dive. We hope you feel well-informed and ready to tackle that quadratic bottleneck yourself.
Until next time.